Hello, everyone, and welcome to a special Stem Cell Agency Facebook Live Ask the Expert. And today our focus is on ALS, amyotrophic lateral sclerosis, or Lou Gehrig's disease, as it's known. We have a terrific panel for you, and we're going to get to those in just a moment. But first, I wanted to say this is meant to be a really engaging and interactive event, and we would like you to post comments and ask questions, and we'll try to get to as many of those as possible. At the end, if you can stick around as well, we'll also have some really important information about clinical trials that I think you'll find very interesting. And so now, without further ado, I'm going to bring in some of our guests, and I'm going to begin with one of my colleagues here at CIRM. There she is, Dr. Lila Collins. She's a senior science officer here at CIRM, and she's going to be talking about some of the research that we're funding. Next, I'm going to go to one of the gentlemen who's behind a clinical trial that we're funding, and this is Dr. Ralph Kern. He's with Brainstorm Therapeutics, and they're running a phase 3 clinical trial that CIRM is involved in. We appear to have lost one of our guests. I'm sure he'll be joining us again in a moment. But the other guest is Dr. Bob Ballo, and he's with Cedars-Sinai. Bob is part of a clinical trial that we're helping fund there as well. And so I think if we could begin with you, Dr. Ballo, if you could talk to us a little bit about the disease itself and also some of the patient experience.
Sure, that absolutely would be glad to. So as you heard, I'm the director of the ALS program here at Cedars-Sinai. I am a neurologist. I see patients who have ALS and take care of them at our multidisciplinary clinic, as well as do research into the mechanisms of ALS. And I work with Dr. Svendsen, who you are all about to hear from soon, if he comes onto the connection as well. And so, I was tasked to mainly actually talk about the disease itself a little bit, and then I'll talk about some of the current strategies we have for treatment, and then lead into a little bit of the research discussion, and then that will take off from there. So as Kevin mentioned, ALS is also known as Lou Gehrig's disease, after a very famous baseball player in 1939 came down with the condition. And as most people on the webinar will know, it's a motor neuron disease, meaning that the motor neurons, the ones that control the muscles that come out of the spinal cord, as well as the ones that are in the brain that control the ones in the spinal cord, are degenerate or lost over time. And that leads to progressive weakness, inability to use the muscles, as well as trouble with something called stiffness or spasticity that comes from damage to the upper motor neurons in the brain. Over time, the disease is progressive, and there are no effective treatments to stop that course. And so people's breathing becomes involved, swallowing becomes involved, and people typically pass away within 3 to 5 years of getting the diagnosis. So it's a very severe disease. It's one that has been mysterious to scientists and physicians for actually about 150 years now. But I should say that it's a very optimistic time, because over the last 10 years or so, the genetics of the disease have really come into focus, and we've learned so much more about how the disease actually happens. And then we have several new approaches that we're able to take to treat the disease that really were never tried or even considered before, and some of which you're about to hear about today, but would have been truly science fiction 10, 15 years ago. So as far as the sort of typical strategies we take now, we do have a lot of things that we do for the patients to help take care of them. We often will have them work in a multidisciplinary clinic, meaning they'll see a variety of providers who are physical therapists, occupational therapists, speech therapists, neuropsychologists, a whole bunch of people who are really expert in managing a lot of the problems that come up when one has a progressive degenerative disease that causes weakness and difficulty with functions, swallowing, breathing, etc. And one of the goals that we put forth when I set up this program years ago and joining Clive here at Cedars was really to involve, if possible, every patient in research. That's one of our mantras. And even if it's not an interventional trial that is going to test a new drug, but something that is going to make progress and bring something meaningful out of that experience for them, in addition to doing our best to take care of everything for them. So we have trials in a variety of things, and I mentioned a few leading into it. But of course, what we're going to talk about today are stem cell therapies. And then actually, our clinic has the good fortune to be involved with both of the trials that are going to be talked about today. And we're absolutely thrilled to be taking those on. So I think what I'll do, Kevin, is pass it back to you. I don't want to take up too much time because I know we want to get into questions, and maybe I'll answer other things as they go along.
Great. Thank you, Bob. And now we're going to go over to Dr. Svendsen, who's going to talk a bit more about the clinical trial that he's running at Cedars-Sinai.
All right, thanks very much, guys, and thanks, Bob, for introducing the topic. We've been working on the specific trial that I'm going to talk about now for approximately 10 years, actually. And what you're going to hear from Ralph next and from myself is a strategy for ALS that's slightly different to what you might expect. Of course, ALS is caused, as you just heard, by the motor neurons dying in the spinal cord and in the cortex. So you have two sets of motor neurons that require movement of the muscles, one in the cortex up here and the other in the back, in your back, in the spine, deep in the spinal cord. Now, ideally, we could take stem cells and turn them into those motor neurons and actually make a new motor neuron. That would be fantastic to replace the motor neuron that's missing. Now, the problem is that it's a very complex process. Motor neuron has to reach out to the muscle and feed it. So the approach that we're taking is slightly different, and that is to put cells in that can protect or stop that motor neuron from dying. And that's two strategies you're going to hear about now. And the nice thing about this is it doesn't require a new motor neuron. And if you go in the stage of the disease where you still have some function or a little function, the way that our approach could actually enhance that function and maybe even stop those remaining motor neurons from dying. The trial that we're doing, funded by CIRM, specifically takes fetal tissue-derived motor neuron cells, progenitor cells from the developing brain and spinal cord, and grows those in a petri dish. And those progenitors can give rise to what we call support cells or glial cells after you put them into the spinal cord. And those support cells go close to the dying motor neurons and wrap themselves around the dying motor neurons and allow those motor neurons, we think, in our animal models, to survive better and function better. When we tried those experiments early on, we didn't see a stronger effect as we wanted to. And so we actually took one more step, which is we engineered those cells to produce a drug or a growth factor, in this case, called GDNF. So now we have a progenitor cell that's going to make this support cell, and we've engineered it to make GDNF, which is a molecule which protects motor neurons from dying in ALS, actually, and in Parkinson's disease as well. These cells were manufactured. The trial itself involves making these cells. We made a huge batch, basically 1,200 vials of this product. That was actually made at City of Hope. The cells are then transported to Cedars-Sinai. This is a one-site trial. And Dr. Baylor and his team have the patient ready. We have a great neurosurgery team here as well, because those cells then have to be put into the spinal cord of the patient. And that does require a neurosurgical technique, and the patient has to go into the actual neurosurgery suite. We then take the cells, we put them in a needle, and we inject them directly into the spinal cord of the ALS patient. Now, this trial is ongoing at this current time. The long-term output, the approval we got for this is called a safety trial or a phase 1 trial. And the FDA, who regulate us, said, you've got to start by going into the spinal cord, low down, where the legs go. So it's called the lumbar spinal cord. So this safety trial is putting the cells in the lumbar spinal cord of 18 patients with ALS here at Cedars-Sinai. The cells we expect will survive in the spinal cord. We're only actually putting them on one side of the spinal cord. And so if there is any effect, although we're not expecting it at this early stage, it's just a safety trial, it'll only be on one side. And the prediction is it may slow down the progression of paralysis in one side only, so one leg. So that's the overall goal of the trial. Once you've had the injection of the cells, you don't need anything else. There's no other treatment. You basically go home. But we do have to give immunosuppression in this trial to stop the cells rejecting. They're not from you, so they're from another source of the cells or some another source. And so we have to give immunosuppression drugs, which allow the cells to survive. And we think after a year, we don't probably need to give those drugs anymore. But for that first year, you have immunosuppression. The trial is on clinicaltrials.gov, or you can contact Dr. Baylor or myself to find out more about it. We closed the trial in terms of information of many aspects of it because this is a phase 1 safety. We can't really prove it's safe until the end of the trial. I can tell you that of the patients we've done, we haven't seen any major complications, and so we currently are very optimistic. It seems to be going well from a safety perspective. And as we go forward with the trial, we'll be releasing more information. So this is an open trial. We are recruiting. We'd be very excited to get more patients involved. So I'll stop there, and I think this is quite a nice transition to Ralph's trial, which is slightly different from the one I just mentioned, but I'll let him take over and explain what Brainstorm are doing.
Okay, Ralph.
Hey, well, thank you very much. I hope I'm on the screen and everybody can hear. It's really a privilege to be here. So I'll introduce myself first. I'm Ralph Kern. I'm the Chief Medical Officer and Chief Operating Officer of Brainstorm Therapeutics. A little bit about myself. I trained in neurology at McGill University, and I practiced as an ALS and neuromuscular neurologist at the University of Toronto. Prior to working in industry, I worked for many years developing treatments for neurological diseases, and I was very fortunate to join Brainstorm about a year and a half ago from Biogen. So it's a privilege to join my colleagues, Dr. Svendsen, Dr. Balo, Dr. Collins today to talk to you about ALS on Facebook Live. At Brainstorm, we're focused on developing cellular therapies for brain disease, and our lead program at the present time is studying Neuron in ALS. And today I'll tell you 3 things. I'm going to talk a little bit about how Neuron works, or how we think it works, what we've learned in the phase 2 study, and the status of the phase 3 study to give you a sense of where we're going. And before I start the details, I'd like to thank and express my gratitude to CIRM for their financial support and guidance. I don't think we would be here today without the forward-looking vision of CIRM. So what is Neuron? So Neuron is a type of stem cell. It's different than the cell that Clive has just described. It's a mesenchymal stem cell that's obtained from the bone marrow through a procedure called a bone marrow aspiration. That procedure involves a needle insertion on the outside of the hip into the bone under anesthetic. It's a brief outpatient procedure, and then the patient goes home. The bone marrow sample is then sent to a manufacturing facility to make a cell therapy for the same person. And in the manufacturing process, stem cells are used to make mature cells that can deliver high levels of molecules, the ones that Dr. Svendsen described called neurotrophic factors. And these are designed to protect neurons and decrease inflammation. So by using one's own cells, we may avoid some issues that are related to rejection or immune reactions against other people's cells. As part of the process, we freeze the mesenchymal cells during the manufacturing so that cells obtained from one bone marrow procedure can be used for 2 to 3 years of treatment. And the finished product contains 125 million cells in 4 mLs. And for those of you that don't do mLs, 4 mLs is about 1/7 of an ounce, so it's a very small volume. And the fluid is contained in a small syringe. It's shipped back to the hospital, and then it's injected back into the patient by a trained healthcare professional in a procedure called a lumbar puncture, where the fluid in the syringe is then inserted into the spinal fluid. And to date, our investigators around the world, and particularly in the United States, have treated over 100 individuals with this technique. So we're getting more and more comfortable with this process. So where are we now in the evaluation of Neuron in ALS? So similar to what Dr. Svendsen described, we've been working on this for a long time. The technology has been in development for 10 years, and we started ALS studies 7 years ago after spending many years learning how to manufacture Neuron. Initially, we did open label studies, and they both showed a slowing of the rate of decline after single transplantation. And based on those promising results, we embarked on a phase 2 randomized study in the US. There was a double-blind placebo-controlled trial involving 48 participants. And the ALS participants were randomized 3-to-1. So 3 people received treatment for every 1 placebo. And it was a single transplantation, and keep that in mind. The study was performed at 3 very well-known centers, the Massachusetts General Hospital, University of Massachusetts, and the Mayo Clinic. And last October, we were fortunate to start the US phase 3 study at 6 sites. 3 of them were the original sites from phase 2, and we have 3 wonderful sites in California, and I will mention Dr. Balo's center, which I've had the privilege to visit, and we're so proud that we're able to work with you in this study. So what did we learn from the randomized phase 2 study? Well, first of all, it was a safety study, so it demonstrated good safety, and it showed good tolerability of the bone marrow aspiration procedure and also the transplantation procedure, and that was very important. We were also able to confirm that the treatment delivered nerve survival factors and reduced inflammation, and as we expected, and we determined that by measuring the spinal fluid before and after treatment. So then what did we see in the results? First of all, we looked at the data 2 new ways that we think are very important. First, we looked at those who were progressing more quickly in the 12-week run-in period to see if there were any differences in the outcomes. And secondly, we looked at how many people had their ALS disease progression stabilize or show signs of improvement. And we call that a responder analysis. In other words, the proportion who showed an outcome of stabilization or improvement. So what did we find? In the total study population, we saw that the rate of ALS progression stabilized soon after treatment in some study participants, and the early improvements decreased over time as expected with a single treatment. And that was pretty much in line with what we were thinking. In addition, we saw an important change in those who were progressing quickly in the run-in period. We could show more stabilization and improvement in that group. And that was also seen for a longer period of time. And in fact, in some of the participants who showed these changes of improvement or stabilization, it could be seen as far out as 12 or 24 weeks after a single transplantation. So essentially what we learned in the phase 2 study was that we would need to do repeat dosing. And we also chose in our phase 3 to study those who were progressing at a faster rate because it would require a smaller study for a shorter period of time. We'd be more certain about the results. We thought that that was the right thing to do for patients with ALS. So where are we now? So I'm happy to report that the study is ongoing at 6 centers in the United States. Including 3 in California, and I mentioned Dr. Balo's site in Los Angeles. It's a randomized study involving 200 individuals, and it's a 1-to-1 randomization of NurOwn or placebo. Participants in the study get 3 doses of NurOwn, 2-month intervals, and I'm very happy to say that the study is going well, that all the sites are recruiting as they should, and we're hoping that we will complete enrollment by the beginning of next year. We included the rapid progressors, as I mentioned, and we also asked that individuals not use edaravone because of the confusion that that might cause in the interpretation of the results. And as I mentioned, we should complete the results and have them by early 2020, and we hope to submit, with a positive result, we hope to submit marketing application to the FDA after the study is completed. For more information about the study, there's a number of ways that you can hear more. One is through CIRM, and we've been able to share information through the various public websites that and facilities that CIRM has in place. You can also look for information on our website, or you can go to clinicaltrials.gov, and be happy to share more information in the question and answer period. Thank you.
Great. Thank you, Ralph. I should say at this point that some people are experiencing some difficulty with the feed. It seems to keep freezing. So we found for some of the people that if you can click out and then come back in, that seems to help improve. Apologize for any technical difficulties. But now we're going to go to Dr. Lila Collins, who's going to talk a bit more about the research that CIRM is funding, but also some of the other programs that are out there. Lila?
Hi there. I think maybe I'd like to first just touch on the drugs that are approved for the treatment of ALS. There are currently 2 approved drugs. The first is called Regenerative, or you may hear it referred to as Riluzole. That was developed by Sanofi. It's been approved for about 20 years. And this is a small molecule drug that protects neurons from some excitation-induced toxicity. And so, as I mentioned, this drug has been approved for a number of years, and it has been found to slow, not yet stop, but slow the progression of the disease, and even improve survival by, modestly, I believe, a few months. And last year, another drug that was developed by Mitsubishi, actually based in Japan, it's called Edavarone. I think it's also known as Radicava. This is another small molecule neuroprotective drug that has been found to be particularly effective in those fast progressing patients. And that was the basis of the phase 3 trial, and the approval was tested in ALS in fairly early stage subjects. And then I think what I'd like to do is touch on some of the other stem cell trials. You've heard from Dr. Kern and Dr. Svendsen about the 2 CIRM-funded stem cell trials. There are some additional trials ongoing in the space. One is kind of interesting, was recently published. This is some work done by Neuralstem. And these are CNS, or central nervous system derived neural progenitor cells that are delivered directly to the spinal cord. And interestingly, there was just a paper published this year on the long-term follow-up from this small phase 1/2A study. So again, a small study, but nevertheless intriguing that after about 18 months to 2 years, there's some promising trends that suggest, improved survival being seen in treated subjects, and also a slower progression of the disease. So we'll, obviously be very excited to see how things continue to develop. And there is also, just starting in Israel, a trial sponsored by a company called Cedars-sinai, and they are developing a human embryonic stem cell derived astrocyte product. Again, similar to the work by Neuralstem in the next trial I'll mention. These are all 3 cell replacement strategies. And in this case, these human embryonic stem cell derived astrocytes will be delivered into the fluid that bathes the central nervous system, the cerebrospinal fluid. And there it's anticipated that they will replace some of the function of the astrocytes that become sick in ALS. And I think Bob touched on this a little bit, but one of the things that we've seen is actually, it appears that in ALS, the astrocytes can become not just sick, but actually toxic to the neurons that they would normally support. So these neural progenitor cells and this astrocyte strategy could potentially replace those astrocytes and help support motor neurons. So that trial is just starting. Additionally, another trial by a company called Q Therapeutics out of Utah here in the States is developing human glial restricted neural progenitor cells. And these cells are transplanted directly into the spinal cord. And the intent there is to replace the lost glial cells, and like I mentioned, the astrocytes, and also could potentially replace the oligodendrocytes. And these are the cells that myelinate axons and help signals travel rapidly. And these may also be lost in ALS. So there's one other trial I'd like to mention. Related, maybe not as advanced as the work that we're supporting through Brainstorm. There's another, just to touch on briefly, another mesenchymal stem cell trial. This is different from NurOwn. These cells are derived from patients' own adipose tissue. And this is an early stage trial that's ongoing at the Mayo Clinic. And I think maybe rather than dive too deeply into all the small molecule trials that are ongoing, I should mention that it's really unique to the stem cell therapies that we have an opportunity to do some potential cell replacement. So this is really an exciting time. Ongoing, there is sort of a mid-stage trial sponsored by Mallinckrodt of an actual gel product that's a neuroprotective strategy that people may be interested to learn a little bit more about. And also Orion Pharmaceutical has an interesting approach to sort of sensitize muscles to the activating signal so that hopefully, one can get more function out of those. And this is similar to a trial that was done by Cytokinetics, although it's a different drug. So we'll see how that works out. So those are really some of the ongoing trials in the space that I just wanted to touch on. Maybe one thing that might be fun to discuss with the panel is, there's been quite a bit of effort in ALS, a lot of different targets explored to try to cure or slow or stop the disease. And maybe one of the things that might be interesting to touch on is, why has it been so difficult to develop drugs for ALS?
Thank you, Lila. And if you're just joining us, welcome to a special Stem Cell Agency Facebook Live Ask the Expert event. Today we're focusing on ALS, and we have a terrific panel. Dr. Lila Collins, as you can see. We also have Doctors Clive Svendsen and Bob Ballo from Cedars-Sinai, and Dr. Ralph Kern from Brainstorm Therapeutics. So I'm going to bring some of the panel in, and maybe they can pick up on what Lila was just asking about, which is, why has it been so difficult to develop therapies for this? I mean, the FDA has just approved a new therapy, and that's the first new drug in 22 years. Why is it such a challenging disease?
I don't know who can start. Can you hear me now?
Are we? Yes. Okay. Maybe I'll start if I think I'm on. One thing that we didn't mention and I touched on was the genetics of the disease, but that's really to sort of get at what are the real upstream causes of the disease. So there are a few examples of trials that Lila didn't get a chance to touch on where we are doing gene therapy approaches for patients who have very specific genetic forms of ALS. That's overall about 10% of patients will have a defined family history, 5 to 10%, where a gene can be identified. And in those individuals, there really are a special group where the types of clinical trials that we're approaching with them are more targeted or more specific. I think what ends up happening for the majority of patients, though, is they don't have a known genetic cause, which to some degree means we don't really know all the events that have sort of incited or caused the disease. And many times as scientists, we've done as best we can to look at all the pathways which are going awry. Many of them you may have heard of mitochondrial dysfunction, reactive oxygen species, glutamate excitotoxicity. I mentioned those last two because those are the targets of two of the drugs that were talked about by Lila, edaravone being an antioxidant and riluzole being targeting for glutamate toxicity. So the problem might be, in part, that these are pathways that are only playing a minor role or are downstream. And so just hitting one of them may not be enough, or it may be too late. So we don't know exactly. Each of them has had, as you've heard, a minor benefit, but I think one of the ideas is that the early causes of the disease really aren't well understood and aren't being targeted by any of the therapies that we're doing right now. So one of the things that's worth mentioning from that perspective is one of the positive aspects of the stem cell approaches compared to the drugs is that they're actually hitting multiple pathways at once. And while sometimes from a drug development standpoint, people don't like that as a thought experiment, it actually has some real advantages because it doesn't necessarily assume that we know exactly the mechanism by how those motor neurons are dying. Each of the cell approaches that you've heard about, either the cell transplantation of neural progenitor cells that Dr. Svendsen talked about, or the transplantation into the spinal fluid of mesenchymal stem cell derivatives that Dr. Kern talked about, either of those are actually protecting motor neurons from a variety of aspects, from a variety of pathways. So I think just to tie them both in, the reasons why we may not be as effective is we don't fully understand the disease, and it's worth taking a step back and considering ways to approach multiple of these pathways at once, at which stem cells are actually one of the better ways to do that currently. So.
Great. Thank you, Dr. Barlow. Perhaps, Dr. Svendsen, if you could join us now and then talk a bit about then how, with all this information, do you then decide which patients to choose? What are the criteria that you use, and how do you come up with that criteria for selecting someone for a clinical trial?
Yeah, I'd like to follow up on something that Bob said, which is we don't know the cause of ALS. And if you don't know the cause of a disease, you have no target to go for. One thing we're trying to use stem cells for in a different way, and another hat that I wear, is a project called Answer ALS. This, if you look on a website, it's called answerals.com, is a project we're collecting 1,000 samples from patients around the country with ALS, and then we make a stem cell line from those samples. They're called induced pluripotent stem cells. And from those induced pluripotent stem cells, we actually can make the motor neurons of each patient in the petri dish. So we can have kind of a spinal cord or a neuron biopsy, if you like, of the patient. And from Answer ALS, we may get some hints as to why those motor neurons in the patients, compared to normal controls, underwent degeneration. And so that may then give us some more hits. And if in order to treat a disease, you kind of have to know what you're targeting. And with sporadic ALS, that 90% Bob mentioned that we don't know the genetics for, we have no clue. So Answer ALS may help provide more targets, and then we can develop drugs to actually interact with targets that we know are affected in the disease. So I think that's the breakthrough we're waiting for. As to subject selection, it's interesting. The two trials you heard about have different criteria for selecting patients. And our criteria in our trial is you have to have some lower weakness in your legs and a score on your breathing. And after that, it's pretty much you can get into the trial. And Bob can go into some of the more details, and you can see again on online at clinicaltrials.gov what our criteria are. But I think each trial does have slightly different criteria for entering, and that's just to enable us to get information from those patients in that specific trial. For instance, we're putting the cells in the lower spinal cord that affects the legs. We're not putting cells in the cervical spinal cord, which affects breathing. So we don't expect to see an effect on breathing, but we do, expect to see an effect on our safety studies based around leg function. The interesting with stem cells is you can target different parts of the nervous system. And the final thing I'll say is on the patient selection and on where we put the cells, as we move our trial forward, we're planning on moving to the cervical cord in the next set of patients, which would be looking at the effects on breathing and arm movement. And then ultimately, I feel that the upper motor neuron is very important in ALS. And I think the Brainstorm trial may be touching on this. They're putting their cells in the cerebral spinal fluid, which may affect the upper motor neuron as well. And the upper motor neuron is, of course, the first neuron that fires when I want to move my hand now. It's firing up in my brain. And so we could completely protect the lower motor neuron, but if the upper one is gone, it's not going to help with your paralysis. And so we're moving towards targeting the upper motor neuron. And that, again, would be a different, perhaps, group of patients that we'd select for that trial. But I think the activity you're hearing on this webinar from Brainstorm ourselves and many other groups that are starting to get into the clinical trial business with stem cells is going to be important. And going back to Bob's point, the nice thing with stem cells is they are what we call pleiotropic in terms of they release lots of things. We don't understand the mechanism. But you know, diabetes, insulin was used in diabetes before we knew the mechanism of why it worked. So sometimes if you get something that works and has an impact on the disease progression, it'll move into, very rapidly for ALS, it'll move along. And then another last thought on why there's been no drugs for this long. I have to say that all neurological diseases, it's been tough to find drugs in the brain. It's because of the complexity of the problem. And so we just have to be patient. I think you'll see an accelerated pace of drug development as we learn more and more about how the brain goes wrong in these diseases.
Great, thank you. Thank you, Clive. Ralph, if we could come to you now and talk about, you're now in a phase 3 clinical trial at Brainstorm Therapeutics. So perhaps you could talk to us a little bit about what you've learned with the first 2 clinical trials that perhaps shaped the way you're now recruiting for this particular one.
Sure. So thank you for that question. Yeah, I think just to also touch on the previous comments, that we've learned that complete certainty isn't required to find a solution. And I think we're targeting pathways. We're not targeting upstream mechanisms. I totally agree the biology, could take some more years to sort out. But if you think of the analogy of multiple sclerosis, there's been tremendous progress in MS without full knowledge of the underlying cause of the disease by targeting pathways. And I think that's what we've learned in the first, particularly in the last study, was that by measuring the spinal fluid and measuring the impact of the treatment on specific pathways such as inflammation, such as the neurotrophic factors, we think that we have an angle on this disease where we might be able to make progress. I think the other important part of it is that we have a very disciplined study protocol and study design, and I think that nothing short of a very rigorous study design can produce results in diseases where there's a lot of variability. And as much as ALS has been well characterized in, for example, the PROACT database, there is some variability from person to person. And the study designs that we have relied on have a run-in period, so we can understand each person's trajectory. We can then look at how that changes after treatment. And we've also been very careful about looking at different types of responses, such as change from baseline, looking at a responder analysis where we see how much of a change we can see in each individual and how much is meaningful. And I think that the combination of both, looking at the biology in the study and linking the changes in the spinal fluid to the clinical outcomes, and having a very disciplined study protocol with the experts that we work with, such as Dr. Balo, really has helped us move forward. I think nothing succeeds like success. And I would say that the next few successes in ALS will bring more share of voice to this disease area. People have been waiting for a long time. I've been an ALS doctor for the longest time, and I can't wait to see 10 successful trials in ALS. I think that's what we all want and what we're hoping for. And I think that we hope to be opening the door to many other people who will then think that ALS is a treatable condition, and we can work together to find really good solutions.
Great, thank you. And I just want to correct something. I put up an incorrect URL a few moments ago. I said it was answerals.com. That was my mistake, sorry. Answerals.org. So hopefully you're able to correct that at home. I want to welcome all our new visitors. We're doing a special Stem Cell Agency Facebook Live event, Ask the Expert About ALS. And we have a question from Lawrence Visaggio saying, why do military personnel get ALS at a greater rate? And I think we've also seen higher rates among NFL players. Is there some particular thing going on here that might give us a clue as to what's happening with ALS? Dr. Kern, you're on screen. Maybe you could start.
Oh boy, I was hoping I would go last, but yeah, no, I think that there have been a number of hypotheses around the higher incidence in military. I think trauma, the common element between the military and football could be trauma. So there is some suggestion that neurodegenerative disorders can be provoked by blunt trauma or trauma to the nervous system, and how that plays into the disease is really unclear. There's some environmental factors that have been cited, such as toxins But again, no one really has identified what those would be. And then finally, there's a question about whether there are some viral causes that potentially could be acquired. But again, I don't think there's been a lot of progress there. So it's surprising that it's higher. It really, we may learn something about the cause of ALS by studying individuals who have had those types of exposures. But I would probably want Dr. Svendsen and Belo to weigh in on this. Hopefully they can do that.
Okay, Dr. Belo, you're up.
Okay. So I think Ralph actually put it quite well. You know, it's likely a combination of factors and it's not fully understood. So one, of course, is actually men are slightly at higher risk than women. They do try to control for that, but there are more men in the military. The trauma and heavy physical exercise or activity is a risk factor as well. And each of these has shown its head before. In fact, a very well-known epidemiologic study showed that Italian soccer players have six-fold increased risk of having ALS. Now, it's challenging to interpret these sometimes because they probably are, again, a mix of factors. Some are environmental, some are genetic, and it's not always taken apart. And sometimes there's something that's basically called observer bias, where in the case of the Italian soccer player study, that was observed for another reason. The study wasn't being done to look at the incidence of ALS. And so a small excess of cases in a small group of people can create a sort of a false positive association. But it does seem to be the case that recurrently, the things that Ralph Kern mentioned, heavy physical activity, some environmental exposures, and perhaps even infections or prior stresses on the system can increase your risk. I always want to point out, and I always do this for the patients, it's important to think about what that risk really means, though. When you talk about the increased risk, another one, by the way, is smoking. So it does actually increase your risk about twofold, which is quite similar to several of the other environmental exposures. What that means is that instead of having a 2 per 100,000 per year risk, one has a 4 per 100,000 per year risk if you're increasing it by double. It means that that's still a very, very rare event in those populations, in the military, in NFL players. It's actually still quite uncommon. It's just, of course, very noticeable. So, you know, as Ralph said, we want to learn from it. There are a lot of really good studies going back, trying to take apart those environmental exposures, correlate them with genetic exposures, and understand it better. And that's important. Unfortunately, I get asked this question a lot from patients, and of course, we can't go back and change things. And it's not even clear that the positive effects of exercise or some of the other things that have shown slightly increased risk don't outweigh the negatives of the risk of ALS on the whole, except, of course, once an individual has it, they have it. So it is a very challenging question. And if Dr. Svendsen has any further insights, I think one thing that he may be able to comment on is ways using iPS cells, which he talked about a little bit, to kind of take apart these genetic and environmental exposures. I think he could comment on that and maybe answer ALS and how that's doing that.
Okay, thanks, Bob. I don't have a lot more to add. I think those were both very good responses to the question. I'll bring up a topical analogy. Genes, I think in any disease, in ALS in particular, genes load the gun and the environment pulls the trigger. What I mean by that is we're all walking around with bunches of genes and we're exposed to different things in the environment. And many people can go into the army or the military and not get ALS. If you're unlucky and you have a vulnerable gene set and you go into the army, you probably will get ALS. Now, what we're trying to understand is, what is that gene set? It's, as Bob mentioned, 10% of ALS patients, we know what the genes are. The other 90%, we're not clever enough yet. We don't know enough about genetics to really untangle. Maybe it's 100 genes which interact. And if you play American football and you have that set of 100 genes, you will go down with ALS. Now, understanding that, again, comes back to modeling how we get to understand the human biology and the human genetics. And the answerals.org, I think I might have said .com.org, is addressing that, as Bob said. We're trying to combine genetic. We do whole genome sequencing on these patients We gather all their clinical information, which includes some epigenetics, i.e., what they do in life and whether they're a football player or in the military will be in there. And then we associate that with both their molecular profile and how their motor neurons react in the petri dish. And that might allow us to understand more this interaction of environment and genetics, which ultimately, I believe, will probably give us the answer to ALS in terms of what causes it. A final thought on that might be one day, I think we're all going to get sequenced. It's under $1,000 now to sequence our genome. You can do 23andMe for $60. That's not really sequencing, but we're going to have a whole genome sequence. It's much more deep knowledge about our genome. So before you go in the military, you may hold up your iPad that has your genome, and they'll check it and they'll say, nope, you're not good for any impact work. You have the wrong gene set. And likewise with other professions for other diseases, probably. So we're entering an interesting era, and we're at the learning phase right now. But going back to the clinical trial, if we had that information on the patient before we enrolled them in the trial, it might be that there are 10 subsets of ALS that would respond. Some would respond to neurone treatment and Brainstorm, and some would respond to the cedars-sinai trial with GDNF. If we knew that before enrolling the patients, boy, that would be fantastic. And then 10% may respond to edaravone, another 10% may respond to another drug. So you can see how understanding the biology of the disease, either through genetics or modeling, is going to really help this kind of personalized medicine that we're moving into.
Great, thank you, Clive. We have another question. This is from Sharon Ward-Keeble, who says, I feel like most of the drug trials ask for the patient to have had first symptoms within 2 years. And that's so hard to define because, as we know, ALS symptoms and a lot of other diseases. We've tried to get my husband on several trials, but he doesn't qualify because of that 2-year rule. So why do we have this 2-year rule? Clive, if we could start with you because you're already on screen, then we'll come to Ralph.
Yeah, and again, I think Bob and Ralph are much better to answer this question. But I'll just briefly say, we have to be sure it's ALS. That 2-year rule is really frustrating because in some ways you want to get in early, but in other ways, if it's something else, a benign fasciculation of some sort that was causing it, or another toxin maybe that you were taking in your food that was causing your problem, you wouldn't want to get, a neurologist cannot give you this horrific diagnosis without being sure. And if you give the diagnosis incorrectly, that's worse than nothing in a sense. I'll hand off to Bob though, and Ralph, because they'll be much more informed on this.
Oh, all right. Bob, I think you're going.
Sorry, yeah. You're up, Ralph. Okay.
Am I on?
Yes.
Oh,
sorry.
Okay, I thought you were just asking for Bob. Yeah, no, it's a tough question. I think that the reality is that all clinical trials are a balance between having as representative a population as possible so that results can be interpreted for as many people as can be, but also try to reduce the variability. And you can imagine that if individuals who were provided a treatment had the disease for 10 years versus 1 year, that would be an enormous difference. The other thing is that ALS is being diagnosed better in my view, and I think people are coming to medical attention. There's more awareness. I think the Ice Bucket Challenge and various other social media campaigns have really raised the awareness of ALS. I think people are coming to medical attention sooner. It still doesn't help someone who's had the disease for 5 years and is looking for a solution. I think from a drug development perspective, the first studies have to be on more narrow populations to reduce the variability. And then as the therapy is shown to be effective, I think it needs to be broadened. And I think other individuals need to see how they do with it. But I think it's just it's the nature of drug development. We looked at all the ALS trials that have been done. There are about 51. And we noticed that the inclusion criteria are really quite narrow in terms of the duration of disease. And maybe I can get Bob to comment because I think as a treating physician, I think you would have a strong perspective on that.
Okay, I think I'm on there. So yeah, I think I don't have a better perspective on the clinical trials. From the point of view of the clinical trialist, it certainly makes sense. We all know that having a more defined group of patients within a certain period of time, and the 2 years is primarily driven by the desire to have patients who are not extremely slow progressors, in fact, that may not change much during the course of the clinical trial. That's the primary driver of that 2-year landmark. If patients have had ALS for longer than 2 to 3 years, and they still meet other inclusion criteria for trials, meaning breathing, being above a certain percentage, et cetera, in a sense, that already defines them as being in this very slowly progressive subset of patients. And it's not that we want to exclude those patients in any way, as Ralph said. It's that we can't include them in a trial that's only doing measurements over, say, 6 months or at most a year if they're not going to change significantly during that period of time. So that's what's the driver of that. But the question being asked is one that I very commonly get, and I certainly understand, and we want to involve as many patients as possible. So in the trial that Dr. Svendsen talked about and is being done here at Cedars for injection of stem cells into the spinal cord, we are a bit more lenient in terms of patients can have had disease up to 3 years. The reason for that, and again, each trial has to be focused on what its primary objectives are. For us, it's primarily safety and some outcome measures, which wouldn't necessarily be dependent on other things. It's mainly leg function, as you heard. So it does vary a little bit, but the primary reason is that I should say that once a drug is approved, perhaps through a given pathway, a given subset of patients, in general, it's approved for all patients. And therefore, while patients can't be involved in the clinical trial, they can still benefit from that drug once it is approved. It's not that they are generally excluded from that. There's a big caveat to that, having said it, which is that recently with edaravone, we saw an interesting situation where the FDA approved the drug for all patients with ALS, but many insurance payers were refusing to pay for the drug unless they met the inclusion criteria of the trial. That's actually new ground for us in ALS. That was never been a bar that's been held before. Obviously, this is only the second drug approved in over 20 years. But for example, riluzole had inclusion criteria which were relatively narrow, and the insurance companies have never held patients to those criteria before. So a lot of that's actually being driven by cost of drug. So I know that's sort of another discussion, but an important point. And the last piece is what we try to do for patients who want to be involved in research but are unable to be involved in interventional trials is make available to them other trial or research opportunities. Being involved in Answer ALS, for example, where they give their stem cells and their DNA and they take part in some outcome measures to really understand the course of the disease so that their experience and their course is going into a database that's going to help us change how it's treated in the future. And in the meantime, there are these other trials going on a more of a subset of patients that they still could benefit from in the future. They just can't take part in the actual trial itself.
That's great. Thank you, Bob. You raise a really interesting question, and it's something that we get asked a lot about here, which is that notion of the right to try. Why can't I test something, particularly for a disease like ALS, which is really challenging and has such a kind of a really rapid progression in many people? Why don't I have the right to try it? And obviously the recent legislation passed by Congress, the right to try, has given many people this kind of this desire and this feeling that this is something they should be able to do now. Ralph, I know that you've had at Brainstorm that this is something that's come up quite recently. So maybe you could talk to us about some of the experiences you've had there.
Sure, happy to. So as you know, the world is changing very, very rapidly, and the amendment to the Food, Drug, and Cosmetic Act, the right to try provision, really has changed our world. We're not 100% sure how that will play out yet, because the interpretation of the law is always something that needs a little more time. I had the fortune of giving a lecture at Columbia University to a bioethics class on the right to try, and I had tried to explain to them how complicated the issue was. And I found at the beginning of the class that it was very polarized. By the end of the class, we all understood that there's a middle ground of individuals who need help, and we need to still have a system to support people and provide therapies and test therapies. So we realized that it's a very complex issue. We spent a lot of time listening to patients, their families. We listened to politicians and legislators. We also listened to and had conversations with the regulatory agencies and many interested individuals. And we realized that in order for us to be able to participate in a Right to Try program, we had to have four key principles. The first is that because the treatments require a certain amount of expertise and hands-on experience, we would only provide this at centers that were involved in our clinical trials who had this experience with the cell therapies, the acquisition of the cells, and also the readministration of the cells. So that was one key principle. And I've been involved in other EAP programs before where we've held that principle. So it's not unique to Brainstorm. The second is that in any program of any type, patients and their families would have to be well educated. We have to have an informed consent. The benefits and risks would have to be explained in detail. And we wouldn't want anyone making an uninformed decision. So that was the second principle. The third was that the law very clearly says that it's only applying for people who are ineligible for current investigational treatments. It has to be an investigational treatment, but also it's not for people who can participate in clinical trials. So we would want to make sure that the inclusion criteria would be met so that people would not be eligible for a clinical trial. And we would never want any program of this type to adversely affect the phase 3 study, which can benefit the broader ALS community. We want to make sure we have a scientifically valid study. We wouldn't want anything to get in the way in terms of resources, focus, and that's a sacrosanct principle. And finally, this is the most difficult issue. This is the one that we couldn't find a solution to, is that because regenerative medicine treatments are fairly expensive, we would need to seek alternative funding solutions in society and to be able to provide the treatment, not just for those who could afford it, but for all ALS patients. And that's something that we couldn't solve. And the reason we decided not to move forward was primarily from an ethical perspective, the equity principle, and the realization that we could not just simply provide the treatment for those who had the resources, and we had to find other solutions. So I think there's a brisk debate in our society. There may be solutions that come up. I know there's other companies looking at Right to Try in other diseases, and we're part of society, and we'll listen to what wise people say, and some of the think tanks, as you know, are thinking through this. So we're open to listen and learn, and that's been our experience so far.
That's great. Thank you, Ralph. Tyler, if we can come to you, one of the things that I get asked a lot when I go to give talks about CIRM and stem cell research is, why do we need placebo control groups for the disease like ALS? Why would we even have a placebo control group? Why is that important?
Hi, right. Yeah, well, one of the challenges that we've touched on today is, that ALS is so variable. And another thing that's constantly changing, or not changing, as fast as we'd all like, is the standard of care can change. So there are good databases. I think we heard earlier about the PROACT database of, I think there it's over 1,000 patients data in there. So we have an idea of what the history of ALS looks like. But at the same time, new drugs are coming on the market, and maybe those patients weren't taking, say, a Regenerative, or they weren't taking edaravone, and maybe patients in your trial now are. So it just including a placebo is a way to really convince yourself, that your therapy is really making an impact against patients that meet those exact same inclusion criteria, on the same standard of care, and diagnosed the same way. And so I think that's really why it's important. But understand how it must be very frustrating if you were looking to participate in a trial, of course, everybody's going to want the active arm of that trial, or to try the treatment. So one trend that addresses this, and I think Ralph may want to touch on this, is the idea of maybe an open label extension to a trial where if you were in the placebo group, you might have an opportunity to receive the therapy after the initial period of the trial is complete, or perhaps a crossover design where you might be receive the placebo for the first, say, year of the trial, and then have the opportunity to receive the active drug after.
Great, thank you, Lila. We're coming to the end of our time now, so I'd like to kind of go to our guests and get some closing thoughts. And Bob, we began with you, so if we could start with you, and just some quick thoughts to leave our viewers with as we go.
Absolutely. So, thank you for inviting me to this. And I guess my closing thoughts would be, as I started off with, that really it's an optimistic time, that really we're talking about things at this webinar that weren't even conceived of 10 years ago, and actually putting them into clinical trial and testing out whether they work. Gene therapies, drug therapies, and stem cell therapies that we talk so much about. So, for patients to engage, to learn, to maintain optimism, and that's what we try very hard to do in our clinic itself, is just to focus on all the things that we can do and the things that we're looking towards doing. And that's basically what I would leave it as. I think we feel very privileged to be involved with these trials and to go forward with each of these things.
Great, thank you. Bob Barlow from Cedars-Sinai. And now, Clive, if you could have some thoughts.
Yeah, just very happy to be on this call webinar. Encourage patients, that as we described, there are no good animal models of ALS, and it's very difficult to understand the disease. So encouraging patients to participate in clinical trials is really important. I know it's very tough to decide even which trial to go in, but I think if you can get involved with a trial and help us to understand this devastating disease, just opening, understanding what's behind these doors that we're all trying to open is very important, and takes it forward for the next set of patients who will understand, hopefully, and work ourselves towards a valid treatment for this horrific disease. So, and I'll just put a shout out and thanks to all the patients who have been involved in these trials already.
Great, thank you, Clive. And Ralph, if you could give us some thoughts on what you're working on and where we hope to go next.
Yeah, so we view this as a journey, that we're not going to give up till we find a solution. And we can't do this without the help of experts like Dr. Svendsen and Dr. Balow. We can't do this without the support of CIRM and other important organizations. And we certainly can't do this without the help of all the patients and their families who are working so hard in the clinical trials to help us find a solution. So we're going to stay at it. We're going to work hard. We're going to find a solution together. And I think the time for ALS is coming, where the biology is becoming well enough understood, and the technology solutions. I think what you heard today are really amazing technology solutions, which were unheard of in the past. The fact that we can actually do these things and test them in a very disciplined way means that I think we're going to be seeing some progress over the next little while. And we just thank everybody for joining in today to hear our story.
That's great. Thank you, Ralph. And finally, Lila, back to you. If you could give us some closing thoughts from your perspective.
Hi, yeah, I would agree. It's really exciting time now with, I think, as Bob mentioned, cell and gene therapy is really, starting to become a reality. So, let's stay tuned and continue to work together. And, I want to echo the thank you to all the patients that have participated in trials, and also to Doctors Balow, Svendsen, and Kern for joining us and for all your amazing work.
Great. Thank you, Lila. And so as we end, I'd just like to thank our terrific panel for some thoughtful and engaging and really informative discussions. That's Lila Collins, obviously from CIRM, doctors Bob Ballo and Klaus Svendsen from Cedars-Sinai, and Ralph Kern from Brainstorm Therapeutics. I'd also like to thank Arlene Hernandez and Joel Arnold at Cedars, who really were instrumental in helping make this happen. They've been great in partnering with us and making this happen. And finally, I'd like to thank you for watching. And just remember that as soon as this broadcast ends, it gets archived on the CIRM, the California Institute for Regenerative Medicine's Facebook page. So you can find it there if there were things that you missed, or if you have friends or you know someone who'd be interested in watching it in its entirety. And lastly, I want to talk about some clinical trials. We've talked here about 2 great clinical trials, and Lila has told us about some other really interesting research that's also in a clinical trial. And the research we fund here at CIRM has to go through rigorous testing and kind of reach very high standards before it's even given the go-ahead by the FDA to be tested in people, because these are in many cases the first time we've even tested this kind of approach. Not all therapies out there that are being offered by clinics are quite so rigorous. And so on our website, if you go to CIRM, www.cirm.ca.gov, you can see it there on the screen. And one of the first buttons on the page at the top of the page is for patients. If you click on that, you'll find a wealth of information about CIRM, about stem cell research, about ALS and some of the clinical trials we're funding, but also about clinical trials and also the information that you need to know, questions you should ask before deciding if a particular trial is appropriate for you or is even a kind of a well-designed one that will help you. And so with that, I'd like to thank you for joining us, and we'll see you again next time.